Hello everyone, Dr. Suresh here, your Games Academy faculty for Biochemistry. So as a part of our Fmge case-based questionnaire for the session July 2023, here is another case-based question. This question is mainly related to the blood glucose estimation. Like what are the blood glucose, the methods for blood glucose estimation and what the type of the bacteria is used for the blood glucose, the blood collector for the glucose estimation and what is the importance of particular type of bacteria in blood collection for the estimation of glucose, and when you collected the blood after the performance in the lab, separation of centrifuging the blood like plasma and serum, and which is the one to make use of for estimation of glucose, that also we'll all discuss along with this case scenario. So a 44-year-old male is admitted for the routine workup. Attending intern collected 5 ml of blood in plain vacuum trainer, remember, at 8 a.m. for various organ function tests, including blood sugar. Time of sample processing was 11 a.m. So 8 a.m. blood has been collected, and 11 a.m. is the time for processing. So 3 hours. Okay, that is the duration where blood is allowed to stand, and the report was received at 12 noon. All laboratory results were normal except blood glucose, which was 42 milligrams per deciliter. So here, 42 milligrams per deciliter is like a hypoglycemic condition, but the patient is normal, right? So what happened? So because the time of blood collection is like 8 a.m. and the processing of the blood at 11 o'clock, so 3 hours. What happened? Otherwise, 42 mg per dl is a hypoglycemic condition. So, consultant biochemistry requested for repeated blood sample in correct vacuum. So, here the mistake has been done collecting the blood in plain vacuum. Why? Because see, the vacuum plays a major role. We are all aware, if you want to estimate the blood glucose, we have to collect the blood in a particular vacuum which prevents metabolism of glucose. As metabolism of glucose proceeds, the amount of glucose will be reduced. So that is a case happened here. So we'll see the possible questions along with this. Yeah. The question related to be asked here, what is the reason for the low blood glucose values in blood collected in plain vacuum? The probable answer is low glucose that has been collected, low glucose in blood collected in plain vacuum is spontaneous glycolysis. So we are all aware glucose will be metabolized by glycolysis, and the key enzymes are there which process the glucose to produce energy. So here the spontaneous glycolysis is the reason behind the low blood glucose values. So because we collected the blood at the 8 a.m. and processed 11, so 3 hours is a sufficient time to process the glucose via glycolysis. So blood should be collected in fluoride vacuum trainers for estimation of blood glucose. So here is again key point, fluoride vacuum trainer. What do fluoride with the glycolysis? So if you have studied the regulation of glycolysis or inhibitors of glycolysis, fluorides are the inhibitor of some enzymes in glycolysis, that is enolase. Enolase is one of the enzyme where substrate level phosphorylation takes place to provide ATP to complete the process or processing of glucose, right? So here this fluoride inhibits the enolase and stops the loss of glucose from the metabolism. Okay, so here blood should be collected in fluoride vacuum as it has got anti-glycolytic agent. Okay, fluoride is acting as an anti-glycolytic agent, and which enzyme it inhibits in glycolysis? It inhibits enolase. The enzyme enolase in glycolysis inhibited by fluoride ions, and sodium fluoride, which is acting as an anti-glycolytic agent here and prevents the loss of glucose by spontaneous glycolysis in collected blood. So that is the reason why if, there is a low blood glucose levels when the person collected the blood in plain vacuum, because it doesn't have any anti-glycolytic agent, so glycolysis happen and most of the glucose is processed by glycolysis. Yeah, this is the gray colored vacuum which is a sodium fluoride coated vacuum, which is the standard one, which is the recommended standard vacuum for the estimation of blood glucose. Plain vacuum is totally supposed to be avoided when you are willing to check your blood glucose levels. So what is the rate of loss of blood glucose if blood glucose is collected in plain vacuum container? So as I said, 3 hours duration, 8 a.m. blood has been collected and 11 o'clock processed. So 3 hours duration. So rate of loss of blood glucose is 10 milligrams per deciliter per hour. So per hour 10 milligrams. So 3 hours duration, the blood was withstanded. So almost there is a loss of 30 milligrams per deciliter. So that means 30 plus 42, it was about 72. The person is normal, is in conscious state, so is not hypoglycemic. So here main thing to explain here, the rate of loss of blood glucose is 10 milligrams per deciliter if you collected the blood in plain vacuum container. And it has to be multiplied with the hours. If 3 hours, 3 into 10, so 30 milligrams of deciliter of glucose will be processed by spontaneous glycolysis if you have collected the blood in plain vacuum container. Again, what are the types of vacuum are being used in various investigations? So when you go to the phlebotomy, like you see different types of colored vacuum. So just now we discussed gray color vacuum is for mainly meant for collection of blood to estimate the glucose. Right. Same way, purple, red, green, and blue are there. So what is for purple? So purple one is the ADTA coated vacuum trainer, which is mainly used for CBC estimation and then glycated hemoglobin estimation. And whereas red one, which is mainly used to collect the blood in this vacuum trainer for major biochemical parameter estimations and also serological studies. And the green one, green topper vacuum trainer, that is mainly is the heparin coated, which is mainly used for the estimation of electrolytes and also ABG, arterial blood gas analysis. And the blue one, which is a uh, sodium citrate coated vacuum trainer, which is mainly used to study the coagulation parameters. So these are the different types of vacuum trainers. So what is the parameter, what is the substrate for the estimation of blood glucose? So here serum is the plasmizer. When you collected the blood, when you centrifuge the blood, it gives you two parts, plasma and serum. So here you might have confused. So serum glucose or plasma. So plasma is the preferred sample compared to serum for blood glucose estimation, as all the criteria for blood glucose report interpretations are laid down based on the plasma values. Moreover, serum is extracted once the blood is clotted, which takes few minutes. This delay in separating serum from blood alters the value of glucose. Hence, plasma is preferred over serum for the blood glucose estimation. So, what are all the other biochemical methods for estimation of glucose other than the automation? So, there are like in general first year medical students might be knowing when they enter into the laboratory for performance of blood glucose estimation. So, they are like glucose oxidase peroxidase method, God-Pod method, famously is like GOD, POD. GOD stands for glucose oxidase, POD stands for peroxidase. And Folin move method, chemical methods or hexokinase method. This is again enzymatic method. Glucose, GOD, POD, hexokinase both are enzymatic method. Folin move is a chemical method. So the most common method in our routine diagnostic lab is like semi-auto or fully auto method, so like GOD, POD method. So that is the enzymatic method. So that's all the facts which were supposed to be aware related to the blood glucose estimation. Hope you understood the concept. That means what is the disadvantage of collecting blood glucose in plain vacuum trainer and what is the type of the vacuum used for blood glucose, accurate blood glucose estimation, or what is the correct vacuum for accurate blood glucose estimation, and what are the different types of vacuum and what are the uses and what are the other methods. So that's all for today. Thank you.